I introduce Professor Dr. Rizia Pretorius, Professor of Physiological Sciences at Stellenbosch University, South Africa. She will talk about the role of microclots and platelet hyperactivation in long COVID thrombotic endothelialitis. So welcome, Dr. Rizia Pretorius.
Thank you very much. Thank you very much. Can you see my full screen?
Yes.
Great. Hello, everyone. It is an enormous privilege to join this wonderful initiative. Thank you for the invitation to share our research endeavors. Research and collaborative discovery science are always done with a dedicated team of researchers and collaborators and postgrad students, too many to name, but a few to point out. Prof. Datel, my long-standing collaborator from University of Liverpool, central to all our work done on microclots. My Stellenbosch team, Dr. Chantel Venter, she's the blood lab manager, and just a few of my dedicated students, Dr. Arne Kruger, Massimo Nunes, Simone Turner, Tom Usher, and Nell Thompson, and Maxine Waters. And then at the bottom there, just some of our funders, who we are very grateful to. My lab and our collaborators from various labs over the world have been identifying inflammatory molecules in circulation that might be involved in and drive pathological clotting. And we have also focused our research endeavors on studying the effects of increased circulating inflammatory molecules and how they interact with cells of the hematological system. We focus in particular on platelets and red blood cells, as well as on the clotting protein fibrinogen. We are also interested in identifying novel inflammatory molecules that might play a role in the persistent symptoms in long COVID and also in the CFS. We are particularly interested in platelet signaling and the role in abnormal clotting. There is a complex relationship between the receptors on platelets and endothelial cells where circulating molecules may bind to. As we now know, damaged endothelial cells and platelet hyperactivation are central pathologies in both long COVID and ME/CFS. And our research has shown these pathologies in various published papers. We are also interested in pathological blood clotting involving the main clotting protein fibrinogen, which is a soluble protein in circulation. We have shown that fibrinogen molecules may become amyloid in nature when they bind to inflammatory molecules such as the spike protein and many others. Fibrinogen then takes on a fibrinoid structure, and we have shown this with various amyloid protein markers like theoflavin T and amitrackers. We call these fibrinoid plasma proteins microclots. Our research therefore focuses on pathology of platelets and endothelial damage markers and microclots. In other words, we study vascular endotheliitis that results from a widespread platelet activation and microclot formation and their various interactions. In 2021, we established the term microclots as a term for the tiny circulating clots consisting of fibrinogen that has become amyloid, and they also entrap various inflammatory molecules. They are present in all inflammatory diseases with an underlying clotting pathology. A unique characteristic of microclots in acute COVID and long COVID, however, is that they are fibrinolytic resistant, which means that they are resistant to clot lysis or clot breakdown. We discovered the fibrinolytic resistant nature of the microclots when we planned an experiment where we looked at proteomics of healthy plasma versus type 2 diabetes, acute COVID and long COVID. Due to time constraints, I will not rehearse the experiments here, but please see our 2021 proteomics paper, where we discuss a double enzymatic digestion method using trypsin. During the data analysis, we discovered that amongst others, there are alpha-2 antiplasmin present, also von Willebrand factor, complement proteins, and various fibrinogen chains that were trapped in the solubilized fibrinolytic resistant microclots. Here is the clotting case cascade, and this is where alpha-2 antiplasmin acts upon, where it prevents clot breakdown. Since 2021, lots of new discoveries followed, not only in our lab, but also in labs of others. In a recent Blood Advances paper from the TOE lab in the UK, the team used our amyloid marker theoflavin T, as well as proteomics, to look at microclot composition in sepsis. You could just imagine that we were very happy to read in their conclusion, and I quote, "Overall, we believe that this is an important step forward in the potential utility of this assay, the microclot assay, as a clinically useful and practically measurable biomarker to identify sepsis, predict DIC and mortality in the ICU." I'm sure you all enjoyed the fantastic presentation that was just before me by the Wust Group. In their 2023 Nature Communications paper, they also found amyloid deposits in muscle using our methodologies. If we agree that ME/CFS is also a post-viral disease, perhaps there are lessons to be learned from acute and long COVID. The focus of our research group in the past months have therefore been on long COVID, but also on ME/CFS. Now, I'm sure I don't need to show this to everyone, as you will know this. But as we focus on the talk is long COVID, here is just some of the worrying thoughts about patient numbers, and I think it's always necessary to show it again. My specific concerns are that some of these patients may progress to ME/CFS or COVID-induced ME/CFS at least. Taking a conservative estimate of 5% of individuals suffering from this long-term consequences, it gets us to a numbers of in hundreds of millions. This is not great news. Back to acute COVID and long COVID. We have looked at blood clotting in acute COVID using the scanning electron microscope, and we found that platelets are damaged, clumped together, and attached to red blood cells. The clotting protein, as I mentioned, that's supposed to be soluble, forms spontaneous microclots. In the micrographs on the far left, you can see platelets that are hyperactivated. Note the yellow arrows. In the middle are micrographs where platelets are attached to red blood cells. Note the white arrows. And on the far right, you can see micrographs of spontaneously formed microclots that even glue together red blood cells. Note the blue arrows. Now, with this ultrastructure in mind, and remember this is acute COVID, we looked at platelet-poor plasma and exposed it to our fluorescent marker theoflavin T, which bind to the open hydrophobic areas in proteins that are damaged. This marker was first used to identify amyloid protein in brains of Alzheimer's disease patients. However, around about 2016, our research group, together with Doug Kell, discovered that theoflavin T also bind to misfolded fibrinogen and fibrin proteins during pathological blood clotting. Here you can see the differences in platelet-poor plasma structure in acute COVID, where we compare plasma smears of acute COVID, that of healthy and diabetic patients. Here you can see the microclot demarcated as the green signal from theoflavin T, our marker of misfolded protein. We found small areas of theoflavin T signal in plasma of healthy individuals. You can see that in the micrographs on the left. More abnormal microclots can be noted in patients sampled with diabetes. What was, however, quite significant to us was the extent of the abnormal microclotting in the acute COVID plasma seen on the far right. Also note the scale bar, 10 micrometers. Early on, we realized that we needed to quantify our platelets and our microclots as viewed under the fluorescent microscope. In our experiments, hematocrit samples were exposed to two fluorescent platelet markers, CD62P, which is the pinkish signal, and a marker for P-selectin. And that is found on the membranes of platelets, but it can also be found as a circulating free molecule. We also used PAC1, which is the green signal that identifies platelets through marking the glycoprotein 2B, 3-alpha on the platelet membrane. As microscopy results are difficult to quantify, we suggested a microclot and platelet grading system based on the various stages of severity. And here's examples of such a grading system. The micrograph plate on the left and the top row shows example of platelets from a healthy or control individual with minimally activated platelets. We demarcated this as stage 1, and then the plate shows increased platelets spreading and the beginning of clumping. And also our micrograph plate on the right are microclots that perhaps the first two rows represent that in a healthy plasma sample, and the clots below a progression into long COVID with severe microclotting in long COVID. Here is a visual to show how platelets will progress through activation, spreading and clumping. This is from the courtesy of BioCo Technologies, our Stellenbosch University startup coagulation pathology lab. You can see the ranges from the left smallest platelet when it's hardly activated and progressing towards all the various stages of significant activation. Here is an example of a really sick individual with long COVID as seen in the right micrograph. We also looked at long COVID whole blood samples using scanning electron microscopy, and here's a representative of, on the left, a healthy platelet, and on the right, of a few platelets that are significantly hyperactivated and damaged. Here is an example of a red blood cell of a healthy individual versus a few glued together by microclots in an individual with long COVID. Again, a larger magnification of a red blood cell showing significant microclot deposits on the red blood cell membrane. This will most definitely impair erythrocyte flow capacity and their ability to transport and deliver oxygen and remove CO2 in the capillary beds, also in the muscle. Here is just an example of a microclot using scanning electron microscopy in an individual with long COVID. Note the scale bar of 4 micrometers. Interestingly, microclot formation is not only found in COVID and long COVID, as I mentioned before, but also in many other inflammatory diseases where clotting pathology are presented. This circles back to recent findings from the Blood Advances paper that showed microclots also in sepsis. We crowdfunded to obtain an imaging flow cytometer, and here are some of our experiments. We are very happy to report that our first long COVID and control microclot imaging flow cytometry method was published last year, and we hope that this would be a useful way to quantify microclots easier in pathology laboratories. Just some of our mean area data and objects per milliliter data showing significant differences between controls and non-COVID samples. Together with Chantal Venter, my blood lab manager, we looked at platelets using a Balvi-funded Metasystems automated system. This system works with our Zeiss fluorescent microscope, and the paper is under review currently. Here are a representative Metasystems fluorescent micrograph of one of our long COVID patients where platelet activation can be seen and platelet spreading as well as clumping in long COVID. So we again used our platelet markers PAC1, which is the green fluorescent signal, and CD62, which is P-selectin, here shown as the orange fluorescent signal. We looked at a 2 by 2 centimeter area. This is scanned automatically, so no cherry picking from our side to choose what we want to photograph. And the system takes 54 images over the area. We believe that the automated method is highly repeatable. And as I mentioned, this is also under review. Just briefly what we did, together with our data scientists and Henry Pretorius, we looked at the Metasystems data. Here you can see a setup where we divided the sizes into 14 size groups. So this is platelets, only platelets. So if we look at group 1 and 2, that probably represents typical non-activated platelet in healthy individuals. Group 1 to 3 probably includes slightly broader ranges of normal platelet sizes, and additional groups then represent the varying degrees of platelet activation up to the very big clumped or hyperactivated platelets. Here you can see bar plots of platelet counts by size groupings for control samples and for participants with long COVID, and a significant difference between the two. And here is a parallel coordinate plot of size groups versus counts of platelets from controls, which is in the green, and long COVID in the purple. Now, previously we published a paper on microclots and fluorescence in MECFS. Massimo Nunes, my PhD student, have just completed a study where he looked at these samples also using the flow cytometry method in his MECFS samples, and he looked at long COVID, MECFS, and controls. Also using THT. Here are examples of the microscopy method, and then we also used our method that we developed for the imaging flow cytometry. We coined a new term, clotometry, but because image flow cytometry is supposedly looking at cells, however, imaging flow clotometry instead looks at clot formation. So here you can see some of our latest results. We found that the number of microclots per milliliter is still the most in long COVID, but that MECFS is between healthy individuals and long COVID, And that is what we have expected, and that's also what our previous results showed from our microscopy that was published last year together with Massimo. Here are the distribution of log transformed object counts within specific area ranges from flow cytometry analysis across healthy participants, ME/CFS and long COVID. So the density plots overlaying the scatter plots show the distribution of data points with medians indicated by the line within the boxes. If we look at panels, A is the count 0 to 100, B is the microclot count 100 to 400, and C is 400 upwards to 900, and then the next one is 900 to 1,600, and then above 1,600 counts. The counts underwent natural logarithm transformation to accentuate underlying patterns within the data to enable us interpretable visual displays. So log count medians are displayed. Now, previously, we did the analysis on the microclots using the microscope. We also embarked on a proteomic study, and this is still under review, also from Massimo. Some interesting results that we got when we looked at the microclots that we digested using proteomics and mass spectrometry is that complement, platelet factor 4, are also significantly dysregulated in ME/CFS. This is basically what we saw with a little bit of changes in the long COVID population as well in our first and our second proteomics analysis. We are also currently looking at E-selectin signal in ME/CFS using fluorescent microscopy. So E-selectin is a marker for endothelial damage. Our results suggest that there are significant endothelial damage, circulating debris, endothelial debris inside the blood samples of patients with ME/CFS. And here you could just see one of those debris parts of the analysis. We could not even take the micrograph over a single screen. We had to take more than one screenshot. We've also been looking at vascular endothelial growth factor, or VEGF. So these results are not yet published. We are still working on it. It's just some preliminary results. VEGF plays a nuanced role in clotting pathology and endotheliitis by influencing endothelial cell function and integrity. Here you can see VEGF on endothelial cell debris at the top and entrapped inside the microclots. Note the red arrows. The presence of elevated VEGF levels generally points towards conditions where there is increased need for angiogenesis, vascular repair, as well as where there are underlying inflammation. So this fits in with our picture of both ME/CFS and long COVID. My postgraduate student, Tom Asher and Anel Thompson, are busy with a biofilm tracer and AmiTracker analysis. As I mentioned, AmiTracker is a similar marker to THT. It also detects amyloid protein. So biofilm might be involved and entrapped in these little microclots as well. And we thought to develop a method for microscopy for that. We found it in our microclots. However, Anel is just currently busy with some of our analysis on our imaging flow cytometry as well. So here is a gate where we found biofilm signal in a long COVID sample on our imaging flow cytometer. The green dots are the microclots positive for biofilm. And these results will be hopefully ready soon to share in a preprint and a paper. Although not the focus of this talk, briefly, something on enzymes that may break down microclots. Nattokinase is a well-known enzyme with clot lysis properties. Many long COVID patients have had great results with this over-the-counter product that originates from the Japanese food natto. Together with my collaborator, Doug Kell from the University of Liverpool and his postdoc, Justine Quixie, we have formulated nattokinase in the University of Liverpool GeneMill. Here we show images of fibrinoid microclot formation and their removal. That's now in the lab, not in patients taking the medication, but in lab, where we added nattokinase to fibrin. Thrombin was also added, and fibrinogen that was incubated together with theoflavin T, and we used LPS to induce microclots. We know that LPS is a well-known amyloid clot inducer. And we imaged after 6 hours on a citation 1 instrument. So in A, you can see fibrinogen with LPS, B, fibrinogen with PBS, and then C with the recombinant nattokinase. Just preliminary results may give us insights on why patients using the product, nattokinase over-the-counter enzyme, might report that some of these symptoms are relieved and alleviated. I'm starting to wrap up. So just a few final summary points. Fibrinolytic microclots are widely present in all chronic inflammatory disease studied to date, and also in healthy individuals. These diseases might include Alzheimer's disease, Parkinson's, rheumatoid arthritis, infection with SARS-CoV leading to acute COVID and long COVID. They can be stained with fluorogenic dyes, such as theoflavin T and amitriptyline dyes. They are commonly around about the size to 2 to 200 micrometers, and they might be resistant to normal processes of fibrinolysis, some even resistant to trypsin, depending on their molecular content. They can also be induced in vitro in both whole blood and platelet-poor plasma with a variety of substances, including LPS, various products, lipoteichoic acid, beta-estradiol, SARS-CoV S1 spike protein. We showed that in various publications. Serum amyloid A is also a molecule that can induce them. They can also exhibit considerable structural and even spectral heterogeneity, reflecting the molecules that were bound to the fibrinogen before its polymerization. Ultimately, microclotting and endothelial damage points to a heterogeneous and complex disease pathology. It is not only simply a size and numbers of microclots present in healthy versus long COVID patients, or for that matter, in any disease with circulating inflammatory molecules, but it is their content, it is their activity, it's their biochemical characteristics that are of importance. We cannot ignore the role of the hyperactivated platelets. Ultimately, microclot formation and platelet hyperactivation are the main driving forces of a thrombotic endotheliitis, and we believe that that can explain most, if not all, of the symptoms that are found in individuals with long COVID and perhaps ME/CFS as well. With that background, we have some exciting next steps. We have various commercial endeavors to try to roll out imaging as well as normal flow cytometry and microscopy microclot testing all over the world. We have some pathology labs already doing it in the US, in Europe, and hopefully we will be rolling it out on a wider scale. Ranges of microclot presence are desperately needed. We need to understand what are the ranges in varying inflammatory diseases, amongst other diseases. We need to figure out what the ranges are. We need to do more.
Please come to an end.
Thank you. I'm nearly done. More proteomics analysis are underway. We need to write that up and understand what is happening inside the microclots. We are identifying more inflammatory novel markers, endothelial cell debris markers, biofilm presence, VEGF, that are entrapped inside the microclots. That could give us an indication of how the microclots act. We are hoping to see clinical trials in the US and the UK on anticoagulation and perhaps with nattokinase. And we are working with various collaborators to formulate grant applications. But mostly, we are involved in awareness. Clinicians need to be made aware, researchers need to be made aware, and we hope that what we have been doing together with our other wonderful collaborators have done just that. Thank you very much, and with that, I give over for questions. Thank you.
So, wow, interesting, Professor Pretorius. Thank you very much for your presentation. Yes, let's start with Q&A, and the first question is, what kind of helpful blood markers can practitioners already perform regarding microclotting?
Thank you. That's a very important question. I think one of the important blood markers that can, or a suite of blood markers that can be asked for currently is VEGF, endothelial markers, damage markers, as well as platelet hyperactivation markers, perhaps P-selectin or things like that. So I think it's very important for those individuals that do not have the ability to have microclot tests done, or even like the analysis that Rob Worcester's been talking about. Those are not easily available for patients, but I think there are lots to be done with normal cardiovascular suites that are available at pathology labs, but mainly endothelial marker damage.
Okay, thank you. So next question. As of today, do you recommend testing for clotting to patients outside of scientific studies?
Outside of scientific studies, I presume in pathology labs, yes, I think that is our focus. We wish to assist pathology labs not only in South Africa, but all over the world, to be able to implement the clotometry method with flow cytometry. I mentioned that we have developed the method on imaging flow cytometry, and the reason why we used imaging flow cytometry is to show that what we are seeing are not cells that by accident slipped through in the analysis, but it is platelet poor plasma with microclots that have been marked. So that is why we use the imaging flow cytometer. However, we do recognize that most, if not all, typical pathology labs don't have an imaging flow cytometry. We are working very hard. We've got certain collaborators going with Beckman Coulter and other such instrumentation groups, where we, together with Bioco Technologies, our startup company, we are trying to get the method validated and used on, and working on a basic flow cytometer, and that will be available to all pathology labs. So hopefully it will be in pathology labs sooner. We are working hard to have awareness on that.
Thank you. So then we move on to the next question. Do you think the microclots endothelium damage is the root cause of long COVID and ME/CFS, or a downstream symptom?
That is a very good question. The root cause of long COVID and ME/CFS is the virus that caused it initially. So in case of long COVID, it is the spike protein and the inflammatory molecules in circulation. So it's a downstream effect of a clotting pathology that starts off during the acute phase due to the presence of spike protein and all the inflammatory molecules in circulation. Spike protein in the acute phase triggers microclot and platelet hyperactivation. It's got receptors on platelets as well as endothelial cells. That starts and kicks off a inflammatory reaction in the endothelial layers for systemic inflammation in all of the vasculature, systemic endotheliitis, resulting in a thrombotic endotheliitis in long COVID. And then this is not resolved in a subset of patients that will then carry on with a thrombotic profile. In the case of ME/CFS, obviously it's not spike protein, except if ME/CFS is now started and long COVID has progressed into ME/CFS. In the case of ME/CFS before acute COVID, it is perhaps because it's post-viral disease, some of the effects of the virus or other components that have induced the symptoms in the first place. So we think it's starting off with the viral components, resulting in a kickoff of endothelial damage, together with microclotted clotting pathologies and widespread platelet activation.
Thank you. So then, which medication works against microclots the best?
So I'm not a clinician, so I would try not to say too much about medications. In the hands of qualified clinicians that would treat patients for platelet hyperactivation and microclot formation, an anticoagulation regime is used with great success. And our collaborator in South Africa, Dr. Jaco Loubser, has been doing this. In the UK and the US, there are various clinicians that use such a regime. And then some patients have had great results with over-the-counter nattokinase, as I showed previously in my slides, and also serrapeptase. However, some patients with serrapeptase might have their MCAS become a little bit worse. Lumbrokinase is also some of the enzymes that we have noted that patients say have a positive effect. However, that said, we cannot really say anything about treatment regimes until we have done proper clinical trials. That is the next thing to do, and I think you will hear from Ray Duncan, who will speak later today on processes that she has in place for such trials.
Okay, thank you. So we have time for a few other questions. So, which diagnostics are available to determine whether a patient has microclots? Could a low perfusion index and visible veins in upright position be an indicator?
I do believe that can be an indicator. Some of the techniques that clinicians use without the presence of a fluorescent microscope or an imaging flow cytometer is something like the EndoPAT. I think one of the previous researchers alluded to the use of that. One of the previous talks, just previously in this session. EndoPAT looks at endothelial function. Another simple test to be done is nail capillaroscopy. That is to look at the capillaries in the nail fold. Some long-COVID patients and ME/CFS patients also have reported that it seems as if their nail fold capillaries are very light colored, not as bright pink as you would expect. So unfortunately, there are limited methods, but the most important methods to use by a clinician is things that look at endothelial efficacy, how efficient is the endothelial layers functioning, like the EndoPAT, and then widespread, well-known pathology tests like the VEGF or those type of pathology marker tests. Unfortunately, that is the only thing currently that's available to patients and clinicians. We hope that soon we will have more pathology labs using a regime of pathology testing for microclots.
Okay, thank you. I think one last question. Is a normal D-dimer proof for no microclotting, or is it not sensitive enough?
It is not a proof at all. So if we look at the definition of D-dimer, D-dimer is a breakdown product of clots breaking down. So you would find it in various pathologies that clinicians know well, like lung embolism. You would find clots breaking down, and then D-dimer as one of the methods to test for clots being present. Unfortunately, when a patient has microclots, the microclots are not broken down as they should. They are prevented to break down because they fibrinolytic resistant. So they will break down eventually, but not in a major tempo. So D-dimer is not a good marker when you are on no treatment. However, we have noted when patients go onto anticoagulation therapy or they take even nattokinase, then in some individuals, their D-dimer do go up after the treatment. They also complain that they feel a little bit sicker during that period, and that can be well explained by the D-dimer now then showing up as a clot breakdown product. So D-dimer, I would not go with D-dimer at all, because it's simply not what we want is to measure. It doesn't work.
Okay, so thank you very much for being with us and your wonderful presentation and insights. Our time for Q&A has come to